Hi everyone, sorry for the delay in getting these videos uploaded onto Halo so you would be able to look at them, but hopefully you'll have time to look at them. We're going to cover in this PowerPoint chapter 23, which is the second of the genomics chapters in your book. And the first one talked more about processes involved in determining the genome of an organism, that is what the DNA sequence is. In this chapter, we're more looking at the functional significance of that in terms of what do genes do, what proteins do they make, and what do those proteins do, and ways of kind of comparing genomes from different organisms in terms of how similar or how different they are. So that's what we'll be doing today. So again, functional genomics is looking at the roles of the genetic sequence and what that gene does. The proteome is all the proteins of an organism. So proteomics is understanding the roles of every protein. And then the final thing they talk about in here is bioinformatics, which is kind of the wedding together of all this information about genomes with computer programs that can do quick analysis and comparisons of genomes. One very useful tool that has been developed is called DNA microarrays, which are also called gene chips. And those are little, kind of like the size of this is a microarray here, of a microscope slide. And they might be made of glass, some of them are plastic. And I don't know how well you can see there, but they have thousands of little tiny spots on them. And the little spots represent DNA sequences that are loaded onto the slide. So it's kind of like little tiny dots of DNA sequences. And then they are like probes because they look at DNA or genes that are active and would attach to the different spots as indicating which genes are being transcribed in a particular cell, in a particular tumor, in whatever case the sample might be from. So, so the DNA fragments are either made synthetically or they're from a sample then amplified by PCR, spotted. So one slide can have tens of thousands of different spots. And it's only about the size of a postage stamp. So it's kind of amazing, the technology of having that many different samples. And so the computer knows the location of each spot and what DNA sequence that spot represents. It's kind of like an inkjet printer or like your TV, the pixels, where you have so many different pixels coming together. So that's kind of the development of the chip itself. And then what you do is, so here you have your microarray. And so we would just be looking at a tiny portion of it on this with 6 different spots. And again, there would be thousands of spots on a real chip. And so here is, if you're looking to see what proteins, for example, are being, or what genes are being transcribed in a particular cell. So you would have the mRNA of from that cell, which would have the genes that are being transcribed in that particular cell. You would use enzymes, etc., to create cDNA. We haven't really talked about cDNA, but cDNA is the DNA that represents the functional mRNA with all the introns removed. So then you kind of hybridize your microarray with your cDNAs, and they will attach to, oh, I should mention that the DNA in the microarrays is single-stranded. And so these are also going to be single-stranded. So they will hybridize together if the other strand is present. So like you have this piece of DNA that represents a particular mRNA that's being transcribed, and if that is being transcribed, it will attach to the corresponding DNA sequence on the well. There's usually some type of fluorescent label or something that can be detected that's added to it, that you will see different colors based on whether or not that gene is being transcribed. And so that's done by like a laser scanner, not by naked eyes, because the spots are too small to see. So this is a pretty amazing technology. It can look at gene expression in different cell types, different things that can turn on different genes by having cells exposed to different hormones or whatever, then see what influence that has on gene expression. One thing that it's used a lot for is tumor profiling. So different types of cancer cells grow and divide and have different characteristics. And so by studying which genes are being expressed, whether it's in different cancer cells, you can maybe localize the types of treatment a particular patient gets based on the genetics of their tumor. So that's just an idea of some of the things that this technology can be used for. It's pretty amazing. And again, I already mentioned this, that it's primarily looking at determining which genes are being turned on or not turned on. So if a gene is turned on, that means its mRNA is present and it will attach to its complementary sequence on the array. If it's not turned on, it won't have any mRNA and it wouldn't be attaching to that complementary sequence and being detected. There's a variation of this procedure called chromatin immunoprecipitation, or CHIP. And that one, instead of determining which genes are being expressed, it looks to find out which proteins are attaching to specific genes. So we talked a lot about gene regulation and it being influenced by transcription factors and other types of regulatory proteins. And so these proteins would attach to specific sites in the DNA. And so in this case, they use antibodies to precipitate the protein that you want to look for. And then you can see where in a particular cell that protein would attach. And so that would be showing you which proteins might be involved in controlling transcription of specific genes. So it's kind of looking more at regulation of protein transcription rather than just whether it is being transcribed or not transcribed. And this is just the technique. You don't need to know about the details, but it's similar to the microarray we talked about. Instead, it's using proteins as what is going to attach to a specific sequence on the DNA to look at the relationship between these proteins and gene transcription. There's a newer kind of form of this, and instead of using DNA, it uses RNA. And so by looking at the mRNA that are present in a particular population of cells, you get the same, I'm sorry, that's my grandfather clock chiming in the background. You get the same kind of information about which genes are active, because those ones that are active would have mRNA present in a particular cell. Again, so you can look at gene transcription, different cell types, healthy versus disease, different stages of development, effects of environmental influence on gene activation. Similarly, to the DNA. This actually is, it's a similar process. And again, I'm not going to go through the details. Since you don't actually do any of these in the lab, it's kind of hard to know the details, but it's using RNA instead of DNA sequences. And the advantage of the RNA versus the DNA microarrays, it seems to be a more accurate at quantifying how much each RNA is present, whether there's a lot, a little, none, and it's better at detecting RNA in low abundance. It also can look at, it can also identify 5' 3' ends and where there are introns and exons, which is not something you can do with the DNA microarray because they're not present in the DNA ones. Okay, so that's more functional genomics involving the connection between DNA, which genes are being turned on, and how proteins might be regulating that genes. The other topic, or the next topic that's mentioned in this chapter, is proteomics, which is determining the role of proteins that a species makes. All of your proteins is called your proteome, kind of like your genome is all your genes, your proteome is all your proteins. And again, we make many different proteins than we have genes. And the reason we have more proteins than we have genes is because of RNA editing, alternative splicing, where the same gene can be used with different splicing mechanisms to get different proteins out of the same gene. RNA editing would be removing different introns from the same gene, and then modification after the protein is made, where you can get the same protein being modified differently to have different functions. And so that's why you can have many, your proteome is much larger than your genome. So it's a little harder to figure out. Oh, these are just saying, so the alternative splicing is where you have, I already kind of talked about this, where a single pre-mRNA is spliced in different ways to give you different proteins. So say this is your pre-mRNA, and this is the primary mechanism for getting more than one protein product from a single gene. By cutting out different exons and introns, you can end up with different proteins from the same gene. And then post-translational modification is just enzymatic primarily modifications of proteins after they're synthesized to give them different shapes and properties that can modify their function. And so you can have the same amino acid sequence being modified differently to have different functions. Okay, so to figure out all the different proteins that a particular cell is producing, and not every cell will make all proteins in the proteome. So different cell types make different proteins, like beta cells of the pancreas make insulin, other cells don't. During developmental stages, you make different proteins based on hormones influence, as well as other things that we talked about earlier. So in proteomics, to try to analyze the proteins present in a particular cell, it uses a process first called two-dimensional gel electrophoresis. And so you probably in one of your labs have done DNA electrophoresis, and we've talked about that a little bit. And so this is a two-step electrophoresis. And the first one is usually done more in a column where you would have the cell, which would have many different proteins. It would be run through an electrophoresis which separates by charge. And then you would take each of those different bands, which they're kind of showing here, and then run those bands through a gel electrophoresis, kind of like the one you use with DNA, which separates according to size. So you're separating first into different charge and then according to different size. So you get different spots representing all your different proteins in your cell that you're studying. This is what a real one looks like. It's not quite as nice as the schematic one. And then you can look for proteins that are present in abnormal cells, proteins that are present when that cell is exposed to a hormone or not exposed to a hormone, proteins that are present in cancer cells versus non-cancerous cells. So you can use it that way, kind of like we were talking about with the DNA, where you're looking at gene, but in this case, you're looking at proteins. And then you can do further analysis of the protein spots. So you can remove those spots from the electrophoresis gel, and using mass spectrophotometry, which we're not going to talk about here, you can actually determine an amino acid sequence from that. Okay. So there's also protein microarrays, which are similar to the chip ones, to study protein expression. They're a little harder to do or not as widely used as the DNA ones. The proteins seem to be much more sensitive to the manipulations of making up the microarray, so they don't hold up as well. And the puring, like when you do a DNA electrophoresis, remember how you purify the DNA first by using different steps to remove other materials. It's harder to do that with proteins, so that methodology is still like being worked out. But again, it would be used to look at different protein expression. These would be possible applications. Okay, so the last thing I wanted to talk about in this chapter is bioinformatics. And so bioinformatics is where all this data about gene sequences, amino acid sequences of proteins is kind of put into a computer, and there are computer programs that can analyze this data and kind of look for similarities, differences, etc. Much more quickly, and it's almost impossible to do it by hand. So it can get that kind of information. For example, this is the amino acid sequence of, I think this is phenylalanine hydroxylase, but it's the DNA sequence starting with the first base to the 1251st base. And so that is a known genome analysis of one gene. So you can see it's pretty complicated. But so there are databases that have all these known DNA sequences for many different genes, many different species, and can kind of analyze if you have a new sequence and you want to look at and say, what could this be, or what is this like? You can enter it and get information about that. So you can look at whether whether the piece of DNA you have is a gene. Does it have a promoter sequence? Does it have a terminator sequence? Does it have splice sites? What is the amino acid sequence that it codes for? Is it similar to other sequences? So homologous to other? And if it is homologous to other sequence, kind of what is the evolutionary differences of those sequences in the different species. So that's the kind of thing that these computer files can be. And they can look at a particular DNA sequence in different ways. This is the DNA sequence of the LAC-Y gene in the lac operon of E. coli. And so this is, that's the one that's shown in the other one. It's not phenylalanine hydroxylase. And this is just showing how you can get, through the computer programming, you can get the computer to look for different things in your sequence. So this one, this is a reading frame here that you can see is not sequences of DNA sequences, but they're using to show it as an example. If you had this sequence here, 54 letters, and you wanted to look at them for different things. So you might say, well, find all the English words in that sequence. And so this program did that. You might want to look for words that are in order for a sentence, or things that go the same sequence in opposite directions. And so you can use, by the tools of a computer, you can look for different things within that DNA sequence in the analysis of a sequence. So it can look for certain sequences, it can look for certain patterns. So it can look for promoters because they tend to, response elements, start codons, stop codons, splice sites, polyase sites. So these are the kind of things that where you would have this piece of DNA with a sequence, it can look and see like all these different characteristics to maybe tell you where a gene starts, where a gene stops, if there is a gene that is a coding gene. So that's the kind of things that you can get from this. It can look whether it's homologous. And so homologous genes that are those that are derived from the same ancestral gene. So here's an example of the LAC-Y gene in E. coli and K. pneumoniae. I can never say that. So you can see 78% of the bases in this gene are identical in those two different bacteria, which means about 22% of them are different, and the red ones are the different ones. And so that kind of information you can get from doing these computer analyses. And with homologous genes, there are two subcategories of homologous genes. One are orthologs, and orthologs are genes that are two different species. So this example of, it's the same gene, it's the LAC-Y gene, and we're looking at comparing it between E. coli and K. pneumoniae. And so we're looking at two different species to see how similar those genes are in those two different species. Those are called orthologs. So the two different versions of the LAC-Y gene in these two different species are orthologs. The other type of homologous genes you can have are called paralogs. And paralogs are different forms of the same gene or homologous genes that are within the same organism. I don't know if I have an example of that here. No. I can't go backwards when I'm narrating, but I don't have an example of a paralog, but it would be ones like we have several different, we have hemoglobin, we have myoglobin. So those would be an example within the same species of that there was probably an ancestral gene, and within that species, they were modified to two different types of genes. So that was the difference between an ortholog and a paralog. So again, all these there's several big databases with all this data of genome sequences, start codons, all kinds of things within them. And there's ones of nucleotide sequences, amino acid sequences, protein motifs, gene expression. So these are all available for researchers to be able to analyze different proteins and DNA to try to look at different characteristics of potential genes. One that is used a lot, and I'm not sure if sometimes they have labs in different bioclasses where they use BLAST, which is Basic Local Alignment Search Tool. And if you have a DNA sequence, say you have some unknown DNA sequence, and you want to see, well, what could this be? You can enter into the sequence into the BLAST database, and it would come up with sequences that are homologous to that and how homologous they are. And so I'll just show you an example here. So this is the phenylalanine hydroxylase, and they're comparing the human phenylalanine hydroxylase with other database sequences of this enzyme in various species. And so obviously, since it's starting with the human, it's 100% homologous or identical to another human, but it's 99% identical to orangutan, 92% identical to rat, 75% to zebrafish, 62% to fruit fly. So that's the kind of data you can get with the BLAST program of comparing DNA sequences from different organisms and how closely